Hi everyone, my name is Emily Gosselin,
And I'm Ryan Hestone.
and our presentation is on T-cell proliferation using CFSE and Thymidine Incorporation. Measuring T-cell proliferation is important for understanding how the body reacts in response to an antigen. For example, you can see in this figure that T cells can differentiate to have a variety of different functions, such as inflammatory or regulatory effector functions. Studying the proliferation of these cells can give us a good idea of how the body will react to an antigen. T-cell proliferation assays give us a chance to study an immune response to an antigen or stimulation under different conditions. For example, if you wanted to test therapeutic formulations. The two common assays used for T-cell proliferation are carboxyfluorescence staining and thymidine incorporation. In order to help you guys understand the slides that we're going to present later in this assay, we figured we'd give you a reminder on some of the important definitions for immunology. So first, just the antigen. An antigen is anything that the immune system views as foreign to the body. An antigen-presenting cell is a cell that presents this antigen to lymphocytes specific to that antigen. And of the antigen-presenting cells, dendritic cells are the most efficient, and they are very important in providing both antigen and co-stimulatory signals needed to activate T-cell effector functions. As a reminder, T-cell is a lymphocyte that serves many purposes in adaptive immunity. You can have cytotoxic T-cells that attack and kill infected cells. You can have helper T-cells that stimulate and support function of the cytotoxic T-cells and also support functions of antibody-secreting B cells. Both of these cells are seen in the figure below, where an antigen is presented to a DC and processed and then presented on the major histocompatibility complexes to CD8 cytotoxic T cells and CD4 helper T cells. The first technique that we're going to describe to you guys is carboxyfluorescence succinimidal ester staining, also known as CFSE. The theory behind CFSE is that it's a fluorescent stain that binds to intracellular amines, so lysine residues, other amino acids, things like that. And CFSE, because it's present in the cytoplasm, is diluted with each cell division. So as you can see from this figure, the signal intensity decreases with each generation of cells proliferated. So some things to think about when you're doing CFSE staining. You should add your solution of CSFE to the T cells before you plate them. And also, if you haven't watched the flow cytometry video yet, you should do that because CFSE uses flow cytometry to measure fluorescence intensity in the cell. And again, building on your flow video, there are several levels of gating to sort out the T cells that have proliferated. So you need to pick your live cells using a stain like DAPI or HUSH. And then you need your T cells, so your CD4 positive, CD8 positive T cells, and then lastly you gate for your CFSE. As with any experiment, CFSE requires that you have several controls in place in order to be able to determine whether or not your T cells have actually proliferated. So to study the effect of a therapeutic on a T cell proliferation, several controls must be in place. You would need a media-only negative control. There's no stimulation in this one, so you should have whatever your basal minimal level of proliferation is. You next would have your stimulating factor, and that would include like mitogens that induce mitosis or antibodies against T cell receptors, CD3 and CD28, which are the two co-stimulatory receptors that are needed for T cell proliferation. You'd have another one that would just have your antigen only, and then you would have an antigen and stimulation factor control, which would be your positive control. And in this control, you should have extensive proliferation, so you'll have something to compare your experimental treatments to. And for T-cell proliferation studies, they can be, for CFSE, they can be in co-culture with DCs that are stimulated and present the antigen to the cells, or they can be cultured with antigens and agents to stimulate T-cell proliferation, so these would include the antibodies against the T-cell receptors. So after you run your flow and you have all of your data that you've collected, you're first going to select your desired T cell population. So these would be gating for the live cells, gating for whether you want CD4 or CD8 positive T cells, and then also for your CFSE. And then you're going to use the histograms that you get that have the count versus the CFSE to see how many cells have proliferated. So if you look at each of these histograms, you can see that there are distinct distinct peaks where generations of cells have proliferated, and using those peaks, you can get an idea of what percentage of cells have proliferated past G0 or G1 or whatever generation you're looking at. If you're looking at Josh's paper on the bottom, he chose to do it after the third generation. So it gives an idea of how many cells have actually proliferated. Alternatively, you can report your data as mean fluorescence intensity. So again, if you're looking at Josh's on the bottom, you see MFI among live CD4 positive cells. MFI is just a measure of how much the cells have proliferated. So you just take the mean average intensity from your histogram for the whole plot, and then as you have more rounds of proliferation, your mean fluorescence intensity should decrease.
As an alternative to CFSE, Thymidine Incorporation is an effective method to evaluate T-cell proliferation. In Thymidine Incorporation, radioactive 3H thymidine nucleosides are incorporated into the cell solution with the T cells, so that as the T cells proliferate, they use the thymidine nucleosides in their DNA. So with each subsequent generation, the radioactivity increases because the number of radioactive thymidine nucleosides in their DNA has increased. The thymidine is replaced by uracil and RNA, so only DNA synthesis is recorded. No RNA for any cellular processes is included. Things to keep in mind: coal culture is very difficult with thymidine incorporation because the thymidine would also be incorporated in proliferating co-cultured cells, such as dendritic cells used as antigen presenters. DNA has to be extracted from the cells for this to be measured, so by lysing the cells, you kill them and viability testing becomes impossible. To measure the radioactivity from the thymidine, a scintillation beta counter, which measures the counts per minute of radioactivity, is utilized. Like CFSE, thymidine incorporation techniques also require a set of controls. However, these controls are basically exactly the same as you would see in a CFSE experiment, with a media-only negative control, an antigens and stimulating factor positive control, and then whichever treatments you like. A major difference with thymidine incorporation is that the radiolabeled DNA could be incorporated into dendritic cells in a co-culture with T cells. As a result, T cells used in thymidine incorporation need to be cultured with agents that stimulate T cell proliferation without using the dendritic cells for antigen presentation. Data from thymidine incorporation is taken using a scintillation beta counter, which measures radioactivity of labeled DNA as counts per minute of beta radiation. You can compare this data to your controls and report either as a counts per minute, which is a raw counts per minute, or as a percentage of the positive control. Both give a measure of total proliferation, but do not indicate how many times the cells have divided, so you don't know how many generations there have been. On the left, this figure is raw counts, and on the right, that figure is as a percentage of the positive control. Thymidine incorporation has advanced beyond the traditional radio labeling to instead use fluorescent labels. These are not radioactive, and you don't need to extract DNA. And so there are some advantages to traditional thymidine incorporation. You can use flow cytometry or fluorescence microscopy, and it's similar to CFSE. The higher MFI, mean fluorescence intensity, equates to higher proliferation. This also allows for co-culture with dendritic cells, in that using flow cytometry you can gate for only the T cells. So you aren't, even though you're labeling the dendritic cells, you can filter those out when you do your flow cytometry. So a number of different fluorescent thymidine analogs have been used. So first is the BIRD-U, and that is detected with fluorescently tagged anti-BIRD-U antibodies. You have to use denaturation of the DNA in order for this to bind, so this is not the most premier technique. The most advanced technique is the EDU analog, and that is detected by a fluorescent azide that binds to the EDU and is based on a click reaction. This requires no DNA denaturation in order to measure the labeled DNA. And on the right there, you can see both the VERG-U fluorescent labeling and the EDU fluorescent labeling. T-cell proliferation is very significant in immunology. T-cell proliferation is indicative of an inflammatory response. So common therapeutic goals with evaluating T-cell proliferation are things that would induce self-antigen tolerance. So something that would help treat autoimmune disease by minimizing the inflammatory response could be tested. Something that would similarly reduce immune activity, an immunosuppressive drug. You could also enhance the immune response in order to be better prepared to face some sort of antigen. That would be done with similar like a prophylactic vaccine. And a lot of these goals incorporate T-cell proliferation. In order to have a physiologically relevant model, T cells need to be co-cultured with dendritic cells. Dendritic cells are exposed to the antigen and a stimulating agent, and that activates antigen presentation. And once these antigens are presented by the dendritic cells using the major histocompatibility complex, that activates the T cell effectors. And dendritic cells also present all the required co-stimulatory signals that induce proliferation.
So that's our presentation. Thank you for your tolerance.
Bring any questions to class next week.